So good afternoon, everyone. Thank you for being here. It is with great pleasure that I would like to introduce Professor Katti. He is a professor of radiology, a director at the Institute of Green Nanotechnology at the Medical School of the University of Missouri in Columbia, USA. He's internationally renowned as a leader in the interconnecting fields of chemistry, radiopharmaceutical sciences, nanotechnology, green nanotechnology, and nanomedicine. He has been awarded many times. We have many things to say about him, but we don't have enough time for his lecture, so we will proceed immediately with what he has to tell us today. Thank you very much for being with us, Professor Katti. It's a great honor. Please.
Yeah, thank you, Penelope, for this kind introduction. Also, I would like to thank the organizers of this meeting for this opportunity. I enjoy every time I'm here in Greece. This time it's very special because we are discussing some realistic collaborative opportunities with Dr. Penelope's group as well as with faculty from this institution. So today, what I would do, I will present you some of our findings, specifically in the area of green nanotechnology, as it relates to developing new therapeutic agents with immunomodulatory properties. Why immunomodulation has become so important in oncology, as well as in other diseases, particularly in oncology. We'll talk about that. Just a brief intro about my institution. As you saw, I come from the University of Missouri, which is in the Midwestern part of the United States. Something unique about my institution, those of you who don't know much about this place, is that we have the world's largest nuclear reactor at the university level. We produce almost every therapeutic isotope used for treating cancer patients. And we supply that to almost all the hospitals within the states, as well as some of our isotopes are exported to Europe also. And that's why we have a great many FDA-approved therapeutic agents, as well as diagnostic agents. Ceratec is the first one, which is used for brain imaging. Quadramat is used for bone palliation therapy for bone cancer patients. TheraSphere is used for injecting into hepatic arteries for treating hepatic tumors. It's a product where we encapsulate yttrium-90 into glass microspheres, and it's a great therapeutic drug for treating hepatic tumors. Then in mid-2000 all the way up until now, we have been focusing our efforts on gold metal, gold isotope, gold nanoparticles, with many interesting products we have. So some of that I'm going to discuss with all of you today. The plan is nanomedicine approaches in cancer therapy. I'll touch base why green nanotechnology is important. Then I will describe to you immunomodulatory features that are manifested by these gold nanoparticles. And then connectivity of gold nanotechnology to ancient Ayurvedic medicine. It happened serendipitously because that's the only way you could get into humans very quickly. That's how we got the regulatory approval for treating human cancer patients. And then this led to a new medical modality called as nano Ayurvedic medicine, very interesting name, and which has been approved by the United States Patents and Trademarks Office. So we can use this as a modality now officially. So that's the, so I have a limited time. I don't do timed talks, but I'm going to do it today. So I'm going to jump several slides so that we focus more on the important aspects. I don't need to describe to you the importance of treating cancer patients, no matter whether you're in Greece, US, China, India, Europe, makes no difference. Cancer affects everybody. It doesn't differentiate between income groups. It doesn't it does differentiate a little bit about ethnicities and all that, but the disease is there across the world. So it's a collective responsibility. So we all need to be addressing this. That's why new diagnostic agents, new therapeutic agents, which can do things that current agents don't do, which allow us to do way beyond the standard of care, are always important. That's where we see always collaborative opportunities. We value collaborative opportunities because it brings a whole new perspective to our discovery process. So there are lots of statistics. We can go through that. Cancer is a deadly disease, takes away close to $2 to $3 trillion from the gross domestic product, and it's only going to increase, as you can see on the slide. So we have a collective responsibility. When I say collective responsibility, let's look at what's going on with the standard of care. What do we have? We have radiotherapy, as shown here. Then we have chemotherapy. Both the radiotherapy as well as the chemotherapy, one of the challenges with this treatment is that they actually lead to enhancement or upregulation of NF-kappa B in both these pathways. So patients who are treated with radiotherapy or chemotherapy, if the tumor is not completely eliminated, the body inherits higher levels of NF-kappa B. What does that mean? That means that higher or upregulation of NF-kappa B results in pro-tumorigenic status to the body. The tumors begin to grow faster, or tumor resides somewhere in the body. 5 years down the road, it's going to come back. When it comes back, then the oncologists have no option because the disease would have become drug resistant. That's why, to start with, we need to develop therapeutics while treating different lesions in the body. Those therapeutics must also address suppressing NF-kappa B. That's why immunomodulation has become so important these days. So immunomodulatory therapy is not just treating cancer. It is about taking care of these signaling pathways to suppress the body's ability to upregulate NF-kappa B. That's something we'll discuss in today's nanomedicine approach. One of the aspects we have focused, or I should say, we focused in earlier investigations is the development of gold, radioactive gold isotopes, gold 198 and 199, very good beta emission. They also have immeasurable gamma. That means you can do scintigraphic imaging, you can monitor the progress of the disease, or the more recent terminology, being able to develop dual imaging and therapy agents, which we call it as theranostics. So lots of advantages about working with gold. And green nanotechnology in my laboratory actually came about when we were trying to meet the need to develop nanoparticulate synthetic technologies with minimal or no use of toxic chemicals. Why is that important? It's important because when you're developing nanotechnology-based diagnostic or therapeutic probes, you're dealing everything at the nanoscale. You don't have the luxury to separate the impurities. There's no such thing. Traditional chromatography that we use for small molecules, oftentimes they don't work. Okay, that's why right from the word go, we need to look at synthetic processes that that eliminate or minimize the use of toxic chemicals, so that you don't have to separate the byproducts or the excess unreacted chemical. That's where we discovered that for a nanotechnologist, when you talk about nanoparticle production of metals, say for example gold or palladium or platinum or silver, a metal of your choice, the process involves injecting electrons into the metal d orbitals. If you are able to do that effectively, you produce nanoparticles. Okay, you're taking metals from plus oxidation state +2, 3, 4, 5 to 0 oxidation state. In nanometals, that's the oxidation state. That's why you need injection of electrons. And the way we did, the rest of the world used sodium borohydride or hydrazine or other toxic chemical reducing agents. But we discovered that there are enough electrons in these plant-based materials. Look at this epigallocatechin gallate from tea, mangiferin from gold, or resveratrol from grapes, or eugenol from cloves. They have lots of electrons. So we could use them We could use them in place of toxic reducing agents, such as sodium borohydride. And indeed, it worked very well. As you can see here, it allowed us to convert gold salt into well-defined gold nanoparticles, because the electrons would interact with the high oxidation state gold precursor, and that leads to the production of gold nanoparticles in a very effective fashion. This is a nice cartoon. Oftentimes, such cartoons work at the lab scale. But all these processes, I have to tell you, we have scaled them up to industrial scales, because the technologies have been licensed for large-scale manufacturing, and they are being used for the production of varieties of different therapeutic products. So something important about gold or any metal, for example, metallic nanoparticle, you have surface atoms. So if a particular phytochemical, for example, epigallocatechin gallate or mangiferin or resveratrol, if it has cancer efficacy, you will enhance that cancer efficacy because you can create hundreds, if not thousands, of signatures of individual phytochemicals on one nanoparticle. That's how you amplify the therapeutic effects to such levels that are generally unachievable if you're going through the macro traditional pharmaceutical approach. This is where nanomedicine approach stands out, is very distinctive compared to traditional macropharmaceutical approach. So amplification is important. And early examples included interacting epigallocatechin gallate with gold salt, producing gold nanoparticles, and well-defined gold nanoparticles. We scaled it up. There's a lot of published work. I'm not going to spend much time. Today's the focus is mangiferin, which is a phytochemical that is present in mango skin. It's an anti-tumor agent, anti-diabetic agent, anti-inflammatory agent, can be used for treating different diseases, but it doesn't work as effectively as it is supposed to, because in the body, this phytochemical is not bioavailable. Body's enzymes metabolize this. So if for this molecule to work in the body for treating various diseases, we're talking about taking this particular phytochemical about a kilogram per day. That is impossible, because that leads to toxicity of the phytochemical from the phytochemical. That's why when you incorporate that on gold nanoparticles, it is a game changer in a spectacular way. You'll see that. When we make these traditional nanoparticles, we always switch back to radioactive nanoparticles, because we have to do the biodistribution, where the materials ends up in the body, how does it clear, where does it localize. So this is you can call it as a match to pair to understand the pharmacokinetics of this, which is very hard. We produce the radio equivalent, then we know everything, where it goes in the body, whether it's IV, intraperitoneal, intratumoral, we can understand all of that. So we did the biodistribution of mangiferin, both the radioactive mangiferin as well as the non-radioactive mangiferin encapsulated gold nanoparticles. These two curves relate to the radioactive gold nanoparticles. This one, the maroon colored, is the non-radioactive one. You can see the radioactive one is great. It suppresses the growth of the tumor completely. There's no growth of the tumor, one injection. The non-radioactive one is not bad. We are seeing efficacy. While this work continues, we are doing more preclinical trials and all that, but we focus this one because this is a low-hanging fruit for us. It is non-radioactive, easy to produce. So we thought that we can quickly complete preclinical studies and go to the government and ask for approval for doing human clinical trials. That's what we have done. That's what I want to show you today. Yeah, that's the one, that's the focus. And if you're wondering how the corona is developed through extensive hydrogen bonding network. Obviously, because you have so many hydroxyl groups, they do extensive hydrogen bonding. The corona is so powerful that body enzymes, they don't unzip all of this mangiferin simultaneously. So it does stay in the body for an appreciable length of time to deliver the therapeutic payload to sites where we want the therapeutic payload to be delivered. And this is how it looks like. It's as I said, it's a great corona. Sorry. Yeah. And the size is important. Size matters when you're dealing with nanomaterials. Why does size matter? Size matters because we want to take advantage of not only the receptor-mediated endocytosis, for site-specific delivery, site-specific uptake. We also want to take advantage of the EPR effects. EPR effects, particularly in a tumor domain, you have porosities due to the vasculature. And the porosity size is about 3 to 400 nanometers. If your nanoparticle is smaller than 3 to 400 nanometers, there's a good chance that you'll be able to penetrate the vasculature to localize nanoparticles within the tumor margins. And that's exactly what's happening here. Our nanoparticles have a size that is ideal for penetration across the tumor vasculature. Not only that, the mangiferin, which our nanoparticles are encapsulated with, mangiferin targets laminin receptors, which are overexpressed in varieties of tumors, including breast tumor. This is the picture, which is worth 1,000 words always. These are laminin receptor specific gold nanoparticles. We have mixed our nanoparticles with prostate tumor cells. The images reflect what you see are the images of dark field microscopic images, as you can see here. And when we block the receptors, laminin receptors with antibodies, and then re-incubate mangiferin gold nanoparticles, you don't see mangiferin gold nanoparticles entering the cells. Everything stays outside, which means that this process of internalization or endocytosis of nanoparticles into the tumors is mediated through laminin receptors. This is not happening. This is not some accidental entry of nanoparticles into the tumor cells. We did the biodistribution, particularly in tumor-bearing animals, into tumoral delivery. As you can see here, much of the injected dose resides in the tumor with minimal lymphatic drainage, very tumor-specific, and which is reflected by these great therapeutic efficacy curves, which is dose-dependent, as you can see here, for prostate tumor. We also did a separate investigation for breast cancers. As you can see here, it's the same thing, dose dependency, excellent therapeutic efficacy as compared to the control group. And at this stage, then we started probing. How is it so efficient? Is there any other mechanism? Then we looked at the NF-kappa B. Mangiferin gold nanoparticles suppress NF-kappa B really well, as you can see here. Again, dose dependency in terms of NF-kappa B suppression. And then the simple biodistribution of the body was important, will be important, because this mangiferin gold nanoparticles target the splenic macrophage. You can see the amount of nanoparticles in the spleen. Most of the nanoparticles, by the way, those of you who are not working with gold nanoparticles, I have to tell you that their main mode of clearance is the hepatobiliary clearance. They end up in the liver. That's not a new development. They always go to the liver. But these particles going from the liver to the spleen is an interesting development. This is where we started probing whether these nanoparticles are targeting the splenic macrophages. That's important in immunomodulation. So we did lots of studies about splenic, the macrophage studies. What we did here, we took this ACE cells, mixed them with macrophagic raw cells. Because raw cells express pro-tumor macrophages, which is an M2 macrophage. When you mix these dog prostate tumor cells with macrophagic cells, the tumor cells begin to multiply, as you can see here, lots of tumor cells. Then what we did, we pre-treated the raw 264.7 cells, pre-treated them with mangiferin gold. Why did we do that? Our objective here was to see whether mangiferin gold can transform pro-tumor M2 macrophage into an anti-tumor M1 macrophage. If they did, the next set of experiments of incubating pre-treated macrophagic cells with the ACE cells, which we have done here, that should show considerable reduction in the population of tumor cells, considerable reduction in the growth of tumor cells. That's exactly what we are seeing. So, which means that mangiferin gold is able to immunomodulate. This is the being immunomodulated by re-educating pro-tumor M2 macrophages into anti-tumor M1 macrophages. This is at the cellular level. Okay, it's a very, very nice investigation. Co-culture studies, you can never go wrong. And this is a standard protocol to say whether a drug is immunomodulatory or not. You have got to be able to transfer on the macrophage axis from pro-tumor into anti-tumor macrophages. So that's exactly what is happening here. So the M1 phenotype, once it is in the body, it suppresses the growth of the tumor, as shown here. And when the M1 macrophages are in abundance in the body, then you can do another macrophage. Do I need to do anything here? It's covering my, yeah. Then you can do another investigation. Then you can measure the amount of IL-12 and IL-10. These are the cytokines. IL-10 cytokine is pro-tumor, IL-12 cytokine is anti-tumor. So because MGF ENPs suppress or transform M2 to M1, while at the same time suppressing NF-kappa B, we see a tremendous transformation of depleting IL-10 and enhancing IL-12, which is required for a drug to act as an immunomodulator agent. That's exactly what we are able to see with this product. So lots of reliable biomarkers have been tested to demonstrate immunomodulatory effects of mangaferin gold nanoparticles. Just to summarize that, what I have shown here, once you treat mangaferin gold with the pro-tumorigenic phenotype, the pro-tumor phenotype transforms itself into anti-tumor M1 phenotype, and that reprogramming or re-education is important because there is a cascade of immunomodulatory effects that kick in terms of reducing IL-10, in terms of reducing NF-kappa B, in terms of enhancing IL-12, which we need for an agent to serve as an immunomodulatory agent. So this whole process, around this time, we went to an industry and showed all of this, what is happening. They got excited. They're also excited because if you look at a lot of green technology talked about across the world to minimize carbon footprint, to minimize carbon emissions, and ours qualifies to be a zero carbon emission process because no toxic material is used, there is no byproduct, and no gas comes out of it. In fact, these productions, large scale, 1,000 gallons are done in small rooms, and you don't need to wear even a gloves or a mask. People still do that for protective reasons, but you don't need to. So this is when we went and talked with a company called DNA, which is in India, and they connected our approach to traditional Indian medicine approach, which is called as Ayurveda. Why did they do that? Because that's how they got the approval from the Indian regulatory agency to conduct clinical trials of this drug in human patients. So I have 6 minutes. I think we're doing okay. So this is the connection. I don't want to spend too much time there. And then they produced this in bulk amounts. They did the quality control, all of that. And then before I show the clinical data, it is not an isolated example. The tumor accumulation, which you saw for mangaferin, is probably the highest. But then we have done EGCG, we have phytochemical from pomegranate, we have phytochemical from grapes, and then we have gamma revec protein. So you can see majority of these nanoparticles show optimal to excellent uptake within the tumors. So one size, we know that one size doesn't fit all. Based on the tumor type, we will be in a position to switch from one nanoparticle to the other. So we have varieties of these nanoparticles. How did we do all of this? We did that because of our ability to label these nanoparticles with gold 198. So scintigraphic imaging biodistribution in nanomedicine was not known until the time we showed that it's possible. If you have gold nanoparticle, work with gold 198 to understand this. So then we did the whole clinical translation from small animal production, small animals, toxicity in pigs, to efficacy in dogs, and now we are finally in human patients. And this is the result. Actually, the government allowed us to test this product in human triple negative breast cancer patients. We were not allowed to use it as a single line therapy, and they don't do that. Regulatory agencies don't allow you if it's a new drug. They tell you to do adjuvant therapy. The patient should be given both the approved chemo agent along with our agent, okay, along with our drug. And the comparison would be with those patients who would receive only the approved chemo agent. And that's a pretty nice way to compare. So pre-treatment, the average median tumor volume of those patient population was 3.2 cubic centimeter. Post-treatment, 90 days with the chemo as well as with our agent. This is a trade name for that Mangiferin Gold. You might say tumor burden reduced a little bit, about 10%. Okay, maybe insignificant. But you need to compare these numbers with this. These are the patient population who were administered only with the chemo agent. This patient population did not get a dose of the Mangiferin Gold. So what happened here? Pre-treatment, this was the tumor volume, tumor burden. Post-treatment in 90 days, the tumors doubled, tripled, or quadrupled. The quality of life of patients dwindled. Lots of patients died in this patient group. None of the patients died here except one patient whose death is unrelated to therapy. The lady had pre-existing condition, but the take-home story is this therapy works really well, and the news is spreading now. Now people are coming forward. We are treating some of the patients on compassionate grounds. Another anecdotal example, just one patient. You can see it's a stage 4 ovarian cancer. Pre-treatment, you have data here. Pre-treatment, you can see lots of lesions. Post-treatment, you don't have a trace of the tumor left in the body. And these are CT images, PET images, very conclusive that this therapy works very effectively. And what I've tried to show you that this therapeutic approach works both at the radioactive level as well as at the non-radioactive level. And it's an immunomodulatory approach. It's not a simple cytotoxic treatment, which I explained to you. WHO, World Health Organization, expects 80 to 85% of the global population to use herbal drugs, but there's no science for those herbal drugs. So we are coming up with some credible science so that herbal drugs can be used with reliability and with these are the bottles in which the breast tumor medication is sold. This is the new modality approved by the US regulatory agency. I think I showed you that one. Lots of people to thank. Again, lots of funding agencies. And with that, I think I will close today's discussions. If you have any questions, comments, I'm here. Let's start them. Thank you.
Very interesting lecture. If you have any questions from the auditorium, because online there have been no questions. I'd like to ask a question.
Okay.
What do you think the next step will be in terms of these academic
Well, in terms of the treatment options, there is a global trend that more and more patients are opting to holistic treatments. This is nothing against existing therapies, but existing therapies, let's accept them. They're very harsh to the body. So if you ask me what is the next step, adjuvant therapies is the best way. We can't take out chemotherapies completely, but let's do dual therapy so that we might be able to reduce the doses of chemotherapy to minimize the toxic effects of chemotherapy. Radiation therapy patients, for example, they need to be taking something, what we have. because it suppresses NF-kappa B, which is elevated whenever a patient is treated with radiation therapy. So those are some nice, innovative clinical approaches that we should consider. It's a combination. Yeah, it's a combination. Just mangiferin alone doesn't do it, because mangiferin is not bioavailable. It is metabolized by the body very rapidly. Once you put that on gold, it is very effective, not just because of mangiferin, but gold also participates. It has an innate role in terms of its anti-angiogenesis property, presumably immunomodulatory property too. Yes, you're right. We haven't done the whole course of investigations on resveratrol, which we intend to do. Very laborious, very time consuming. So each step takes a couple of years. So maybe in another 5 years, we'll have more data on resveratrol. Yeah. Yes. Yeah, it's a synergistic effect of phytochemical with gold nanoparticles.